Welcome to Decoding the Data Ecosystem, a podcast with valuable insights, inspiring stories, and practical advice to guide you on your journey through the world of omics research and how the NIH Common Fund data ecosystem can support that journey. I'm your host, Dr. Alyssa Dillman, and I'm the Training and Engagement Director of the CFDE Training Center. My background's in bioinformatics, and I've been working in bioinformatics and data science training and engagement space for almost the past 15 years. I'm really passionate about lowering the barriers of entry for these fields through creative engagement activities such as these podcasts. Now let's get to the show.
Hello everyone, it's your host, Alyssa. This podcast is highlighting one of my favorite events of the year, the BioIT Fair Data Hackathon. If you're not familiar, this hackathon kicks off the BioIT World Conference and Expo and brings together an incredible mix of data scientists, software engineers, students, and life science professionals to tackle real-world research challenges in a fast-paced, collaborative environment. This year's event was once again hosted by us at the CFDE Training Center, marking our second year leading the BioIT Fair Data Hackathon. While last year's event really focused on just introducing participants to CFDE datasets, tools, and resources, this year we placed greater emphasis on interoperability. Teams were encouraged to connect CFDE resources with external datasets, tools, and platforms, demonstrating how FAIR data principles can help bridge silos and enable new discoveries across the biomedical research ecosystem. Over the course of just 48 hours, 6 teams explored topics ranging from drug repurposing and exercise mimetics to cancer prognostics, post-translational modification discovery, and drug side effects. By combining expertise from across disciplines and leveraging interoperable data resources, participants developed innovative solutions that highlight what's possible when researchers can more easily connect and reuse data across systems. This year, we also introduced something new, project awards. Teams competed for a share of $15,000 in prizes, recognizing innovative approaches to improving interoperability and advancing open, data-driven discovery. Even more exciting to me, the winning team was based on a challenge proposed by someone outside of the CFDE community. And to me, this is a powerful example of how the hackathon is expanding its reach, bringing in new voices, new perspectives, and new scientific questions while creating opportunities for collaboration across the broader biomedical research ecosystem. And one of the things I love most about this event is seeing people with different backgrounds and expertise come together so quickly. And participants don't just contribute their own skills, they learn from each other, share ideas openly, and build solutions that none of them could have created alone. And that's really what this hackathon is all about. Collaboration, open science, and creating resources that others can build upon long after the event ends. Before we jump into the presentations, a quick note. These talks were originally delivered with slides during the hackathon showcase. If you'd like the full experience, be sure to check out the video version of this episode. Now, let's hear from the teams and learn more about the innovative solutions they built during the 2026 BioIT Fair Data Hackathon.
All right, everybody. We are ready to start doing our final presentations. So like I said, each team gets 8 minutes, and I will aggressively creep on you once you run over the 8 minutes. We will save questions until everyone has presented. Any final questions before we get started with our brave first team? All right. The judges are back in that table. Yes. Okay. Take it away.
Okay, very good. So I will check for the 8 o'clock deadline. 8-minute deadline. We're not doing an 8 o'clock deadline, goodness gracious. So I will introduce us. We are the team Drug Repository. For repurposing drugs, it turns out our repository became known as the Repository, and we thought it was a nice name, so we decided to stick with it. So I am Pat McNeely. I was in charge of the team. This is the team. I will roll through quickly because we are now down to 7 minutes and seconds. So our background here, we were focused on drug repurposing and also a little bit on novel small compounds that have been tested in laboratory settings and show evidence of possible use, and are presented in the CFTE data sets, but aren't necessarily on the market right now. So why focus on drug repurposing if we already have drugs on the market that we know are safe for one thing? If we come up with evidence that's already in a knowledge base somewhere, it makes a lot of sense to target something that has already cleared FDA approval for new targets. So we focus on target identification, where in this case, we're augmenting our targets. So we start off looking at biomarkers of given disease conditions using biomarker KD. We then included a string protein-protein interaction analysis to pull in protein-protein interactions, look at networks of genes nearby to the genes in question. And finally, we looked to implement real-time scoring. So we started off looking at liver cancer. We actually managed to target 5 cancers live. Hopefully we'll get to show you this demo, where we're providing a potential researcher with the ability to toggle how much they prioritize novel drug discovery, novel gene discovery, novel gene activity, those kinds of things. So here's a high-level view of our pipeline. So we focus on fair data. It's a fair data hackathon. It's a good place to start. We start off with our user input. And then you can see we've got a number of data sources that we sort of built out as the hackathon progressed. We started off with 3. I think we ended up with 6 all told. We move into our pipeline processing, where we've got a bunch of stuff happening under the hood. We managed to convince an AI to convince us that it was doing a decent job. Fortunately, the AI was writing Python and JavaScript code for us, so we could actually look at it, press go ourselves, and maybe be a little bit more convinced of it. We then write the output to disk. We cache things that we download from the CFTE sources, from other data sources, that kind of thing. And then we actually were able to put together a visualization for users to kind of get a better view of what the AI scripts are telling the user, but also to interact with the data on the back end and get a feel for what was recovered from the various CFDE sources. So just to dig in a little bit more here, we started off by building, I mentioned liver cancer, so hepatocellular carcinoma data pipeline, starting off at biomarker KB. We query the biomarker KB API, we get information there. We digest that down into gene symbols. We look into GTEx, IDG, GDC, Links L1000, lots of acronyms for projects within CFDE, and also with GDC, CFDE adjacent. We also, like I said, mentioned, we expanded into protein-protein interactions to sort of expand our net for novel discovery. We were then able to actually implement multi-cancer generalization. So we implemented 4 extra cancer scenarios just to see how far we could push ourselves and the code before either things broke or we decided it was time for the night. And finally, we built that dashboard that I mentioned. So very quickly, because that 7 minutes is calling, I believe the demo is here. So what we are looking at here is a very constrained environment for me to move the mouse. So here we've got our dashboard. At the top, we have ranked drug candidates, and we're giving each of our data sources a weight of 1 on the side. And you can see biomarker KB's contribution, GTEx contribution, IDG. These sliders work in real time. So you can see this slider is updating potential drug candidates that have been sourced from links after we've applied the GTEx, IDG, and GDC filters. So putatively, a user could come in and sort of scroll through the links sourced candidate compounds after filtering through GTEx for the specificity of gene expression, IDG for whether or not that particular gene is well established, and go through the various CFDE data sources based on that filter. Now, as we scroll down, we've got statistics for the pipeline, what was pulled out, information about biomarkers, information about the GTech statistics. I will move quickly because that 8-minute window is closing fast. So I'll close the live demo here just to show we also were able to implement pancreatic cancer searches, colorectal cancer, lung adenocarcinoma, and breast cancer searches. So all of these get pulled in, they're cached locally on the back end. We get a number of interesting potential topics, where we've got novel targets or repurposing targets, as sourced from the IDG and Pharos database. So with that, I will pass it to Anusha. I will not stop sharing because that would be the wrong thing to do. There, we're back.
So in case you were wondering what our future would look like, don't worry, I'm here to tell you. So first of all, we're expanding to more diseases. 48-hour window is pretty narrow. We did not even expect to reach 5 cancers, but we do want to move on to the rarer cancers, maybe neurodegenerative diseases if possible, as much as possible, obviously. And we also want to enrich our string network. We use the confidence score threshold to 700. So I want to play around and see how many more candidates we could get from that. Next, we want to integrate more CFDE data sets, of course. One of them was HubMap, which we were looking into. So right now, GTE is giving us which organ is enriched, differentially enriched from the other organs, and which biomarkers are present. So HubMap would probably tell us which cell type is expressing this expression. So that could be more useful data for the researcher. Then we want to connect our candidates to CAMDL. So this would tell us two things. One is the admit property, which is, if you're not familiar, the absorption, the distribution, the metabolism, and the excretion. So sort of like a toxicity indicator, and also the clinical trial status. Again, helpful for pharmaceuticals maybe. Then we want to package a pip installable tool. No one wants to clone a GitHub repository and then create a virtual environment. So we want to do that in the next step. And of course, MCP. We want to make it an MCP server. So maybe use your favorite AI tool, whether it be Claude, ChatGPT. Just type in your prompt about, oh, I want to run this. I want to know the top 5 repurposable drugs. It will run the tool for you, and then there you go. No need to even run the entire tool. So I would say it was a pretty productive hackathon for us. We didn't expect us to go this far. So thank you so much for listening to us, being patient, and hope you liked that.
Thank you so much. Like I said, we will hold our questions to the end. So let's bring up the next team, which is our side effects team.
All right. So hi, everyone. So this is Team Repurpose RX. We have built a computational pipeline, reproducible pipeline, to reverse engineer pharmacologicals and DPT. So I'm going to just talk about a few stats and then pass it on to my colleague. All right. So bringing a new drug to market is slow, expensive, risky. We all know how long. It takes a long time basically to get a drug out. Costs roughly $3 billion, takes about 17 years, and about 90% of phase 1 candidates fail even before reaching FDA approval. So the stats on the traditional method of doing the drug repurposing, I just gave it, but then with this approach that we have followed in this hackathon, we will be able to get the drug repurposing thing down from 17 years to about 12 years, down the cost by about 85%, and also the approval rate by about 30%. Some of the drugs right now, even with the ones with on targets, they have side effects, and most drugs actually bind to unintended molecules, causing adverse side effects. But with this new approach that we're going to be presenting, we're going to have improved outcomes.
And just so you know, this project focused on utilizing the side effect of each drug to be able to repurposing those API. So side effect is the direct evidence that this drug has a clinical effect on human biology beyond the intended target. And we can utilize this side effect to create a direct path to identify new therapeutic uses. These are some examples.
Right. So here's a history of where this has worked before. There have been cases in the past, kind of most famously this top one here, Viagra. Many people don't know Viagra was actually initially a hypertension drug, so meant to lower blood pressure. But they found that as a side effect of Viagra, wouldn't it be a hypotension? Anyway, as a side effect of Viagra, there were sustained erections in many patients, and so then the side effect then turned into a new indication for this drug. And this has happened for some other drugs in the past as well. For example, another hypertension drug caused hair growth as a side effect, so now it's been repurposed for alopecia. Same thing with a drug that was actually for influenza, for the flu, that was repurposed for Parkinson's disease. So our project replaces this kind of happenstance, serendipity that might be observed after doing clinical trials, and tries to predict those side effects and those repurposing opportunities further up in the pipeline and do it systematically. So we're introducing Repurpose RX, which is our reproducible computational pipeline that uses CFD data to reverse engineer pharmacological serendipity. And here are some details about how our pipeline actually works.
Yes, so in this pipeline, user would input a preferred side effect, and the output at the end is going to be a ranked target dashboard, where the first ranked target would be the most potential target to be repurposed for that side effect. So the pipeline will work for from side effect using the database Drug Central. We would filter for drug and target with the system of, for example, user could choose for drug that only going to contain small molecule drug, high affinity with the target, only in location, etc. From there, you will have like a list of target. And now I would like to rank that target by a target score system. And we have decided to have the score system based on two main category. The first is the target with the most drug that reach to that same target. The higher the drug used to that same target, the higher the possibility of that target could become a new drug repurposing target. But the number of drug is not enough, because drug could be different. Because even though the same drug going to the same target, their mechanism could be different. So we also take into account how similar or different the pathway of the drug reaching to that target. And in this similarity system, we take into account the location anatomy between the side effect and the drug, the pathway type component within the drug pathway, and also different gene compound that is a part of that pathway as well. And user can specify different way to tune up or down how important it is for each categories. And we also use a lot of CFDA fair data pipeline, published API in this pipeline.
So the first example we decided to address was hypotension. So hypotension being a side effect. So certain drugs cause you to have low blood pressure as a side effect. But of course, there are many people out there who have high blood pressure as a problematic disease. And so how can we maybe repurpose drugs that cause low blood pressure as a side effect and actually use them to treat high blood pressure? So we tested our pipeline using hypotension as the input side effect. And I outputted several hits. And this top hit here, if we just look into ADRA1D, which was the top candidate target from our pipeline. And when we look it up, it is in fact a protein that's involved in vasoconstriction, which is a very important pathway for blood pressure. And this actually worked so well that we were thinking, okay, maybe it's something messed up, and the drugs that were used to identify this target were actually blood pressure medications, but they aren't. You can see on the right, these are just some of, there are about 20 drugs used to identify this target. These are just some of them, and you can see that they're drugs that are usually prescribed for schizophrenia, nausea, prostate hyperplasia or urinary tract infections. And so these are, not blood pressure related things at all, but we were able to use these drugs to identify a potential new target for blood pressure medication. Further examples that we have run through our pipeline and are analyzing the results currently are other side effects that could lead to good repurposing indications. For example, cytopenia is a low blood count, so that could be used to potentially treat blood cancers. Same with hyperpigmentation, could be used to treat vitiligo. Libido changes, sexual dysfunction, weight gain could be used to treat things like cachexia and anorexia.
Last but not least, for future consideration, since I mentioned before, user could input their chosen weight, but we want to have an optimal weight. So in the future, we will hope to tune scoring weight with real data, using some model to train with real data to find an optimal weight. We could also integrate additional public data in this project as well. For example, target specific subpopulations so that it could be more optimal than female or male. And last but not least, one we validated on Drug Central for existed drug. We would extend it to drug that is already approved by phase 2 and phase 3, but have been abandoned by different companies to see how potential it could be for repurposing. Thank you so much.
Thank you.
Thanks.
All right, up next, we've got Exercise as Medicine. It's our team project 1.
All right. Yeah, we are the project 1, exercise in medicine as medicine. Well, is that how the project called? I'm not sure. But all right. So, but we rename it like Omics in Motion, right? Because our goal is to compare the Motorpack study on endurance training to any other disease, right? So in this case, whatever data set we could find, that we could do the comparison to see what is changing in exercise relative to what is changing in other diseases. And that's it. All of us, we are going to present on the different phases of this project.
Yeah, it is not.
There we go.
Sorry about that. So our goal is to identify reversal genes, or genes that move in opposite directions in exercise versus disease, with our hypothesis being that key reversal genes will flag the protective pathways and candidate therapeutic targets for different diseases. Our FAIR framework for cross-dataset exercise and disease comparison is built around the Motorpack Rat Endurance Training Omics as the healthy reference paired with public rat disease datasets. For this study, we took in raw expression matrices, normalized expression data, and pre-computed differential expression gene tables, and we generated ranked discordant feature tables, which were gene expression, GSA quadrant visualizations, pathway and CFDE knowledge center context, and an interactive Explorer web app.
So as Rula has mentioned, in terms of the two pre-processing route that we took, is we either consider row counts as our initial data, or we consider pre-normalized FPKM to feed all of these input data into a single harmonization layer that was keyed on ensemble gene IDs and a controlled tissue vocabulary as if the metadata. So our pipeline kind of goes from raw data to doing differential gene expression analysis, and that was using EdgeR. And then we fed this, or we aligned this actually to Motorpack data, and then we perform gene set enrichment analysis and discordance in the aim to generate rank tables, quadrant plot, and other visualization. And in terms of the different data set that we curated, we actually created 6 disease data sets from different disease areas, going from liver disease to looking at type 2 diabetes, looking at carcinoma, looking at metabolic comparison, doing a metabolic conversion, doing a cocaine versus control comparison, as well as addiction, and looking at a proteomics data that concentrates on obesity, type 2 diabetes, and training, performance training. And these were from different omics, coming from different omics modalities, I would say.
So we had a really hard time finding these publicly available datasets suitable for this analysis. In fact, it was the hardest part of this whole project. Here are some of the issues we faced. Unfortunately, we were able to find the datasets that we found. Yeah, so to systematically identify candidate exercise as medicine genes, we developed a discordance scoring framework that robustly ranks molecular features showing opposite direction responses between exercise and disease across matched tissues. By standardizing and comparing exercise and disease-associated fold changes, the framework prioritizes genes potentially reversed by exercise in disease-associated contexts. And we further incorporated a 3-tier evidence system based on statistical significance, effect size, and directional consistency to provide an interpretable and scalable framework for analyzing and selecting biologically meaningful exercise disease contrast signatures across heterogeneous omics datasets. And to demonstrate the framework, we applied it to a multi-tissue obesity transcriptomics datasets and a proteomics dataset. So this heat map summarizes the proportion of genes that are among our top 3 tiers across tissues and data types. Overall, we observed widespread discordance signatures across nearly all tissues, suggesting that exercise broadly counteracts obesity-associated molecular dysregulation rather than affecting only one isolated pathway or tissue. So these quadrant plots provide a more detailed view. Here, each point represents a gene, and the shaded quadrants represent sign discordant regions, meaning that exercise and obesity drive the gene in opposite directions. So across multiple tissues, the liver showed some of the strongest and highest confidence reversal patterns, including several tier 1 genes. And this aligns with the biology because liver metabolism and insulin signaling are heavily altered in both obesity and endurance exercise. And here are some strongest candidate genes that we found, which are involved in metabolic and signaling pathways. And then strong discordance signatures were also observed across muscle, intestine, adipose, and brain, indicating that exercise broadly opposes obesity-associated programs across multiple tissues. Together, these results support that endurance exercise systematically counteracts obesity-associated molecular dysregulations at a multi-tissue level.
Also, rather than just looking at individual genes, because if you remember yesterday, and some of the tissue types, there were no significant genes, for example, in the brain. If you look at them in terms of the gene sets that determine pathways, you start to see some really interesting pathways that are kind of discordant or concordant between the conditions. So I want to focus on this cocaine addiction data set where both exercise and the cocaine addiction were leading to an elevation in dopaminergic pathways, as well as more synaptic remodeling and synaptic signaling and stuff like that. Versus another thing that's interesting is in the cocaine addiction, there were more neuroinflammatory pathways and these were the ones that were kind of reversed due to exercise. So this type of analysis was conducted on all of the data sets that we were using. And so another one is kind of also looking at obesity, where you start to see things like more wound healing and the leaner kind of groups, and as well as muscle regeneration and more related pathways.
All right. Yeah. So with respect to the project, we created. So we made available all the scripts that we used to process and harmonize all the data sets. We also have the scripts that perform the analysis, which are all available. We could not finish without also doing a quick application that allows the exploration of these data sets and this finding. We go disease by disease. You can, for example, select one, and then you go to you can visualize the volcano plots for each of the individual studies, or you can look at the GCA analysis, the results, and you can zoom in each of them, and you can see the tables and all that, right? So we want everything to be fair, so that's why we created this application as well. All right, I think that's all.
All right, so next we've got the PTM PTMs group. Yes. Okay, excellent. All right, and again, this is the, making sure I'm getting the numbers right for our judges, mining the CFDE for post-translational modification data, project 5.
Okay, so today we'll be talking about mining common fund data ecosystem for post-translational modification, PTMs. And PTMs are chemical modifications that adjust functions of protein or may adjust protein function. And we started by doing audit of the CFD datasets and trying to find all the PTMs across the ecosystem and build a database containing all the information about that. And once we get there, let me ask Chase to talk about how we did it.
Sure, yeah. So as Vlado said, part of the work here was mining CFDE for PTMs. And the way we kind of did this, at least our approach, is a couple buddies of mine have been building this sort of like agentic workspace where you could, like, all of us could collaborate in sort of the same environment with the same agents. So that's what you're kind of seeing here. So we'll send some links in the end, and you'll see some QR codes. All of those are also being served from this workspace. And so the general idea is we were hoping to get everybody's expertise in one place so we could mine stuff, we could build dashboards, we could write code, all using the same agent and the same virtual machine, using all the same packages and all that kind of stuff. So this was our sort of joint development environment for everything that we did. And with that, I'll pass it over to Jenna. So we wanted to sort of target, once we sort of did all that curation of all of those PTMs and CFDE, we wanted to try to answer like an actual scientific question that would be relevant to a researcher. I think a lot of times we build dashboards of annotations that aren't actually relevant to the people that we're actually serving downstream. And so we wanted to actually try to answer a question that Jana had around PTMs and how these modifications work in disease.
Okay, so like Chase mentioned, there's a lot of data sets in the CFDE that have these PTM information that as a researcher I cannot use, and I would like to know the disease relevant information from these data sets. So what we did was we built dashboards that based on mining these data sets. And so just as a quick reminder, PTMs are naturally occurring in our body, in the proteins. And these changes in these PTMs, so like increase in phosphorylation at a specific site could be associated with Alzheimer's, or decrease in glycosylation at another site could be associated with another disease. So we wanted to see these differential changes in these PTMs in different conditions or different diseases. Given so first we probed motor pack, since it has like clear baseline versus condition, which is the exercise. So we used the different conditions, and we have the 7 tissue types, different PTMs, which are acetylation, glycosylation, and phosphorylation, males and females, and the 4 time points after exercise. And we were able to create this dashboard. And if you could please scan this QR code so that you can see live updates of our dashboard. So here, this dashboard is on MotorPack again. So let's search the protein that we know is involved in metabolism. So this is a metabolism protein, or important in energy production. So here you could like plug in the different conditions that you want to test, or the different proteins that you want to test. So let's say condition A is female, sedentary, and sedentary the baseline, and condition B is female, 8 weeks. And here we could see this post-translational modification at this site is like phosphorylation is increased after 8 weeks of exercise, which is interesting because this might be associated with activation of this energy production protein that could enhance energy production. So next we also built a 3D structure where we employed AlphaFold to see exactly where this PTM is occurring within the 3D structure of the protein, because we know it might affect the 3D structure, it might affect protein-protein interaction. So we were interested in seeing that, like visualizing it, so you can see this for every protein. We also built a network of like the pathways enriched, and you can see the post-translational modification of this protein and what's enriched, which pathways are enriched for this specific PTM. So we still need to correlate this with perturbations to function and to add protein-protein interaction information. However, we weren't able to do that in 48 hours. So next, as a medical doctor myself, I was interested also in seeing how these PTMs could affect diseases and disease changes. So we know most significantly or most prominently, we know that neurodegenerative diseases have a lot of protein static issues and issues with protein folding and post-translational modifications. So we first started by probing glygen database, which has glycosylation sites, to check for neurodegenerative proteins. And we traced back the word they just said to the original source from PubMed that has that Zygen is feeding to get the exact sources and the data from this PubMed from these sources to be able to identify specific sites that are undergoing PTMs that are associated with different diseases. So we were able to identify almost 2,500 proteins with these modifications, all these glycosylation sites, these PTM sites, and we used novel language. What's
natural language processing?
Novel language processing to be able to make sure that these annotations are accurate. So I'll also ask you to scan this QR code to see our second dashboard. I'll also open it and give you a okay.
It renders better on desktop.
All right, so here you can see all the stats above, all the stats that we were able to identify. And we have this PTM track viewer where, let's say you want to search a protein. Again, we started this with neurodegenerative diseases. So this was first built on 30 proteins and then we expanded. So let's start with neurodegenerative, which is prion protein. So here there's this prion protein that is dysregulated in prion disease. And here you could choose like the condition that you want to study, the baseline, which PTM you want to study. There's only glycosylation and phosphorylation, and the species. And you can compare the conditions. Why isn't it scrolling? Okay, so you can compare the different conditions, and here you see all the PTMs that we were able to identify within this protein. And then you can see the prion like in prion disease, this phosphorylation is decreased at this specific site. And interestingly, we were also able to map all the SNPs, which are mutations causing diseases and or non-causing diseases in the genome. And here you can see the diamonds. If it's colored red, it means this mutation is associated with a disease. And we overlaid it with the PTM positions. And you could see here, for example, that this specific SNP is disease associated, like at this PTM. So we were able to integrate these two data sets to be able to identify novel PTM sites that could be novel PTM sites that could be associated with disease.
Yeah, show the other one.
We're done. The time is over.
Yeah.
1 minute.
You still have time.
Okay. All right, sorry. Okay, I thought my time was over. Okay, so actually we have here, like if you click on this specific SNP, you're able to identify which diseases it is associated with. So you can overlap these SNPs with like the PTM and see these different conditions or diseases that could be associated. And here you see the pathway enrichment for the disease that you could choose, and PTM changes by condition, site evidence, variant PTM overlap. And also, what's also interesting here is that you can also see the specific, it's not, is it showing? Yes, so you can see here the specific drug, like enzyme that is phosphorylating at this specific site, which could be used with the first group that presented the mimetics, and they can like maybe collaborate on trying to find specific mimetics. I don't know, or repurposing. Right. So, oh, that's not our project.
Okay. So this was a team effort. So thank you, Amy, Rebecca, Christina, Jana, and Chase for doing wonderful work and putting this together. Thank you, everyone.
Thank you. Okay, up next we have our Project 3, Visible Neural Networks for Cancer Drug Response, or CellMap VNN.
So basically, when we look at cancer predictions, and by that I mean like outcomes related to cancer. So maybe how long someone survives or how they react to a drug, for example. These things are really difficult to parse due to like the large genomic complexity of like the data we're looking at, right? And then we have a lot of machine learning models that they claim to do a bunch of great stuff with the gene expression data that we have. But when you look at like the clinician, when you look at like the researcher, in order to interpret those like machine learning model results, it takes a lot of time. It takes a lot of time to parse like these like correlations between like specific mutations and specific phenotypes or outcomes. So that is something that we really wanted to target in our project during this hackathon. There are some like existing tools in architecture. So we work with CellMap VNN. This is a neural net architecture that actually mirrors the hierarchy of how proteins and genes are like organized within the cell, and it uses tumor genotypes to predict like various endpoints in cancer. This might be, as I said before, disease prognosis, drug response, etc. So on the right, you can see the cell map itself. It has the organization of the genes and proteins like within the cell clustered here. Okay, this brings us to our research question. Can we actually expand this neural net to, number one, deliver patient-level explainability? So currently, this neural net delivers population-level explainability, which can be sometimes hard to like derive like true like clinical or research hypotheses from this. Can we also integrate multimodal data? Can we like include other like perhaps like clinical covariates? Can we like make the model performance better using this? And then also, can we, as everyone is wont to do, provide a GUI? And can we integrate like some external data to like make sure that this model output is easier for people to explore, to research with, to work with, and to play with, so yeah.
Okay, so we started with a base model, CellMap VNN, and this model is mainly genotype focused. That means it only has the genotype information like the structural variants, copy number alterations, and it output the static explainability file. And as an output to our model, we also added some clinical covariates so that we could make clinical context-aware prediction, and we integrated it with the GUI. So the clinical features that we added in the VNN are the mutation count, fraction genome altered, and the sample type. And these three features are based on what we think is biologically and clinically meaningful in the tumor level context and in the survival prediction. So to test whether the model with the added clinical variables actually improve the model performance or not, we compared the model performance with the clinical variables and without the clinical variables. So on the left, we have this validation loss, and we can see the model with the clinical variables has lower validation loss, which is indicated by the green curve, as compared to the model without the clinical variables. And also we have this validation accuracy curve on the right. And as we can see, the model with the clinical variables has higher validation accuracy, and also it achieved that higher validation accuracy earlier in the process. So the key point here is that the model with the clinical variables overall improved the predictive performance of the VNN that we used.
So now coming to the GUI that we want to explore. So the GUI has 3 dashboards. First one is a dashboard which has like the basic dataset info and all the necessary information we need to like get started with. And the second level has like LLM generated interpretations from all the data which we have in the main hierarchical map. We convert it into something which anyone can see and like, hey, I can use this data and I can interpret it along with my own clinical description. And also like we also have a special map with all the other genome types, other protein structures, how we are linking a specific protein structure to the other top three most important relevant protein structures which may cause cancer, or which is not like which has probability of not causing cancer. Like we can interpret through that. So I'm gonna take you through a project demo on how it's gonna be. Okay, you can take a look at this. So this is like our, I'm sorry, it's like our basic on how our dashboard looks like. Our we are looking at the multimodal data integration. We also integrated the drug genome interaction data set for from the CFD data set on how we are like how the drugs are integrated, like how we can predict the therapy, like treatment therapy. And also like we engineered a custom model architecture on the visible neural network with 3 clinical covariates so that the model performance increase as my friend said that. And also our LLM descriptive interpretations are like we will see in the next one. So we can see how we can most recurrently important nests are across the patients. Nest is nothing but a protein structure, which is like a hierarchical map of it. Like it has 365 protein structures, which probably cause which are proved to be causing cancer for that. So LLM interpretation, for example, I took one patient here and we run it through this and we can see the patient details, how they are like, I'm sorry, like I'm just gonna mute this. Yeah, so then top 3 most important factors, and also like the top genes, how they are going to do that, and also the direction if they worsened it, or how the biological role if the tumor suppression, and how the targeted therapies can be interpreted. And also we will go through the map we were talking about. This is the map on how the protein structures are. So the map also has interpretation on how the clinical reasoning and additional stuff, and also it regulates like we can see the individual genome data with this. I'm sorry. So the key takeaways: we have improved predictive performance and patient level annotation, which is very useful for further research. We have additional data integration throughout the model architecture and result analysis, and we are interactive, interpretable hypothesis platform which can be used by anyone, which is like useful.
Don't worry, guys. Last slide. So, how do we improve? So currently, we only consider one clinical study. We want to integrate more different data sets from different studies through CBio portal. And lastly, we think this can work on different cancer types as well. So we want to test that on different cancer type, and then integrate more multimodal data like RNA sequence or clinical covariates and like image data as well. So that's all. Thank you, guys. Under 8 minutes.
Excellent. All right, we are on to our last but not least team. We've got our exercise mimetics discovery with gene set foundations model, or project 6. Let me just make sure I refresh this. And hopefully that is updated. Oh, I'm so sorry.
All right. Hi, everyone. So our group worked on Project 6, which we're calling XMIME. So exercise is a powerful tool for preventing disease and promoting overall health. However, many people can't exercise for various reasons, injury, illness, or getting older. Despite decades of research, we still don't have a systematic way to find drugs that replicate what exercise does at the molecular level. So most approaches only include a few pathways or tissues, or don't involve large-scale genomics data. So that's the gap we're trying to address with our project. So MotorPack provides a detailed map of how exercise shapes gene expression, and the links dataset catalogs millions of drug perturbations. And our project connects these 2 datasets using a gene set foundation model published in 2025 by the Mayan Lab to rank compounds by how closely they replicate gene exercise signatures. So GSFM is a biological deep learning language model. So instead of predicting words, it predicts gene sets based on relationships from massive collections of gene sets across different tissues and pathways. So this lets us predict complex patterns between exercise, gene signatures, and drug profiles. So we hypothesize our pipeline will identify molecular compounds whose signatures overlap gene-induced gene expression patterns to reveal exercise mimetics. We built a two-stage pipeline to integrate VoterPack and links data using the GSFM, which we will explain next. And our deliverable is a list of exercise mimetic compounds and a reproducible analysis workflow.
Okay, so here's a brief overview of our pipeline, which essentially divides into 6 stages. The pipeline starts with taking data from VoterPack. So we get tissue, rat tissue data sets, gene signatures, basically exercise signatures from 8 rat tissues. And it goes through a signature derivation step, which is essentially just filtering them and selecting the top 250 upregulated, downregulated genes. And since it's a rat data set, we first do the ortholog mapping step to basically convert the gene symbols to a human gene symbol list, so that it's easy to compare them later on. On the other side, we get the links database data set and split them into, again, up and down halves of the gene sets. So both of these go into the gene set foundation models, where every gene set is converted into an embedding vector. And after that, we use these 4 scoring methods to score each compound against every tissue used from VoterPack. Once we have our scores, we go ahead and do the ranking, wherein, because like exercise is a full body phenomenon, it's not like an isolated thing. We first do a per tissue robust rank aggregation. So we basically fuse all the scores and then do the pan tissue word accounts. Later, after the ranking is done, we validate them using 3 different methods, which is a control recovery, wherein we see if the 9 known mimetics actually appear in the top ranked list. Then we do the GSFM ablation to see if the model added anything by its own. And also we do the bootstrap stability testing to see if the top 50 ranks at least are stable across different perturbations. After that, we add a UMAP galaxy plot to see if the clustering of exercise mimetics and the exercise signatures is together or not. And we also finally annotate our compounds with GTEx aging signatures and Ferro's druggability data, and everything finally comes out as a fair JSON LD output.
So we wanted to begin in the first phase to do a quick data sanity check with VoterPack. So we plotted the differentially expressed genes, DEGs, across different tissues and time points. This is to guide us whether we should focus on one tissue or just look at every single tissue as a whole. And we see, like, for example, you see brown adipose tissue. It's consistently in both down and up regulated, the left and right graphs. It has a lot of DEG genes. And this kind of guided us to just look at not just muscle tissues and heart tissues, which are predominant in exercise, but to just look at holistically pan tissue. Also, we scoped down our project to the 8-week time point to just focus on the effects of chronic exercise. And for sure, future work can focus on the smaller data granularity in terms of time points. Okay. Yeah.
So these are some of the top candidates which are we are identified by using different scoring methods. So we have used why we have used these many scoring methods, because every scoring method has their own pros and cons. And so let's say exam, if it can rank a compound with top rank, like rank 1, and other compound other method will rank the same compound with different ranking. So to solve that, we have used robust ranking aggregation method, which can make all the ranks into a single rank, and it will give the final result.
Yeah, here's the final result. We got a very unexpected final result. You see that the lower is the better, right? So the GSF went, the one with the GSF FM, they got the worst result. Yeah. So I've been thinking that for whole morning, and then I think to explain it in very mathematically and statistically ways that because the GSFM has a really large dimension. It's trained on very large model data. So, but the tissues we used like to do the embedding, they're very small. So like it's like 100. But the ESFM has like 10 over 100K. So if you want to calculate the cosine similarity between these two vectors, you get you get a sparse vector. So that's why it didn't do the encoding really well. But we extend our research to the anti-aging. So GSF. So on the x-axis is the exercise limit score. So with GSFM, you can extend. You can add a y-axis. You can combine the exercise mimic and anti-aging axis together, and so you can find those. So those we can find some new compounds, which lie on the upper right. So those are the novelty candidates. So which has very good exercise mimic, and also it also have a good effect on the anti-aging. So that's a really good discovery. Yeah. Also, we made a dashboard. I hope it worked. Okay, so I deployed on the Hugging Face space, and you can see our result right here. Here is and also you can ask questions about that. Okay, so yeah, it's a page. Okay, so let's go. The presentation. Yeah.
Okay, so this is how we followed and adhere to the fair data principles. Everything is public. We use licenses, everything like that. Extended discussion, implications. So with every machine learning model, it's really important to consider what are the limitations, explainability, things like that. One of the things that it's beneficial is that it's easy for us to scale this up and enable experimental discovery, drug repurposing. Limitations, there is a limited amount of human tissue data available in MotorPack dataset, so that's something we should consider. Future work, building more visualization tools, integrating more multiomics and disease data to look at connections with pre-existing conditions, and then integrating pathway-aware integration with GXFM. Thank you. These are our references.
All right, let's give a round of applause for all of the presentations.
You have been listening to Decoding the Data Ecosystem, a CFDE Training Center podcast. Be sure to check out our other episodes in this series. Find out more information about CFDE data and tools that may be valuable to your research by visiting our website.